This video presents a set of 4 experiments used for identifying functional HIV-1 Nef protein motifs and for developing peptide-based inhibitors. Specific short peptides derived from motifs found in full-length proteins both retain their biological function and competitively inhibit the function of the full-length protein. To identify apoptotic motifs in HIV-1 Nef, Jurkat cells are exposed to Nef Scanning peptides, and a tunnel apoptotic assay is performed to identify peptides that induce apoptosis. A second set of peptides containing variations on a Nef secretion motif, the secretion modification region, or SMR, are transfected into cells along with Nef-GFP. Loss of function due to competitive inhibition, indicated by decreased secretion of Nef-GFP, is assessed by fluorimetry. To identify cellular factors that interact with the secretion modification region, or SMR of Nef, co-immunoprecipitations are performed using SMR wild-type Nef as bait. Finally, to test whether these interactions affect secretion, antibodies against identified binding partners are transfected into cells to assess whether they antagonize Nef motif secretion. The results obtained identify 2 functional apoptotic motifs in Nef and an inhibitor peptide that antagonizes a functional Nef secretion motif as well as its cellular binding partners required for secretion.
The overall goal of this set of 4 experiments is to accelerate the identification of functional motifs in target proteins and then use that data to develop peptide-based inhibitors of these functional motifs. This set of protocols has been useful for our group in helping us to answer key questions related to HIV pathogenesis, such as understanding the role of Nef-driven secretion in pathogenesis and in deciphering the mechanisms underlying Nef's ability to manipulate the exosomal trafficking pathway. Today, demonstrating the procedures will be Dr. Mingbo Huang, who is a research instructor in my laboratory.
Begin this procedure with a set of peptides scanning the region of interest. For this experiment, 20 mer peptides with a 10 amino acid overlap spanning the entire HIV-1 Nef protein were obtained from the AIDS reagent program. To identify Nef apoptotic motifs, perform an apoptosis assay using the Nef Scanning peptides individually as follows. Add 1 milliliter of culture medium containing 10 nanograms per milliliter of peptide per 35 millimeter plate of Jurkat cell cultures, and incubate cultures for 24 hours at 37 degrees Celsius. Following the incubation, perform a terminal deoxynucleotidyl transferase-mediated DUTP biotin nick end labeling, or TUNEL assay, to assess apoptosis. To do this, first wash cells with PBS. Then, aspirate the PBS and add 4% paraformaldehyde in PBS. Incubate for 30 minutes at room temperature to fix the cells. After fixing the cells, wash them with PBS and add Triton X100. Incubate for 10 minutes at room temperature to permeabilize them. After fixing and permeabilization of cells, add TUNEL reagent and incubate the cells for 1 hour at 37 degrees Celsius. Following permeabilization, rinse the cells twice with PBS. Then, determine the percentage of stained cells by epifluorescence on a computer-controlled microscopy system. After transfecting Jurkat cells with Nef-GFP plasmid and different variations of the SMR peptide using chariot protein delivery reagent kit, as described in the accompanying document, harvest the medium. To determine transfection efficiency by fluorescent microscopy, capture fluorescent and bright field images, and determine the percentage of cells transfected. Then, to assess inhibition of Nef-GFP secretion, transfer 100 microliters of the cell-free conditioned medium to individual wells of a 96-well black microtiter plate. Use a fluorimeter with excitation wavelength 485 nanometers and emission wavelength 515 nanometers to measure the GFP fluorescence in the medium in relative fluorescence units. After the reading is complete, transfer the data to a PC. Set the level of GFP fluorescence in the control medium at 100%. Then, compare this to the level of GFP fluorescence in the medium from cells co-transfected with various SMR peptides to determine changes in Nef-GFP secretion. The co-IP for isolating motif binding partners is a difficult step. Make sure that the beads are properly agitated during incubations, and that as much liquid as possible is removed from the beads during each wash step. Grow Jurkat cells in RPMI 1640 medium containing 10% FBS at 37 degrees Celsius in a T75 tissue culture flask. Pellet cells by centrifugation at 1000 times G for 20 minutes at 4 degrees Celsius. Resuspend the cell pellet in 1 milliliter of PBS and transfer it to a 1.5 milliliter microcentrifuge tube. Spin at 1000 times G for 1 minute. Then, resuspend the pellet in 1 milliliter of anti-flag lysis buffer by gentle pipetting. Differentially microcentrifuge the suspensions at 2000 times G for 5 minutes and at 13,000 times G for 10 minutes to pellet the cell debris and DNA respectively. Collect the supernatant, hereafter referred to as the lysate. After determining the protein concentration, add 1 milligram of lysate protein and 1 microgram of either the SMR wild type or SMR mutant peptide to 20 microliters of anti-flag M2 affinity gel. This affinity gel is hereafter referred to as the resin. Bring the mixture to a final volume of 1 milliliter in co-IP buffer. Also prepare a negative control in which the lysate is combined with resin in the absence of either peptide. Incubate the mixtures at 4 degrees Celsius overnight with end-over-end rotation. The next day, pellet the resin by microcentrifugation at 5000 to 8000 times G for 30 seconds. Following the spin, wait for 1 to 2 minutes before handling the samples to allow the resin to settle in the tube. Remove the supernatant with a narrow-end pipette tip or a Hamilton syringe, taking care not to transfer any resin. Wash the resin 4 times by resuspending it in 500 microliters of wash buffer, followed by microcentrifugation. To elute the bound cellular proteins, add 15 micrograms of excess 3X flag peptide to the tube in 50 microliters of wash buffer and incubate on a rocker shaker for 30 minutes at room temperature. After the incubation, centrifuge at 5000 to 8000 times G for 30 seconds. After allowing the tubes to settle for 2 minutes, collect and save the supernatant containing the eluted proteins. After concentrating the eluates by acetone precipitation, boil the samples in Lamley sample buffer. Then, separate the proteins by SDS page on a 40 to 20% Tris HCl criterion precast gel. Stain the gel with Kumasi Brilliant Blue R250. To assess antibody inhibition, co-transfect Jurkat cells with Nef-GFP and either anti-alpha tubulin or anti-mortalin antibody using Chariot protein delivery reagent kit as described in the accompanying document. Collect the cell-free conditioned medium from the harvested cells. Transfer 100 microliters to individual wells of a 96-well black microtiter plate. Then, Measure GFP fluorescence in the medium using a fluorimeter with excitation wavelength 485 nanometers and emission wavelength 515 nanometers in relative fluorescent units. After reading the plate, transfer the data to a PC and set the level of GFP fluorescence in the anti-alpha tubulin control at 100%. Compare this to the level of GFP fluorescence in the medium from cells co-transfected with anti-mortalin antibody to determine changes in Nef-GFP secretion. To map apoptotic motifs of HIV-1 Nef proteins, peptide scanning analysis was performed as described in this video. The y-axis shows percentage of cells that are tunnel labeled, and the x-axis denotes the treatment condition. As shown here, two separate regions of HIV-1 Nef were identified that induce apoptosis. These are indicated by increased tunnel labeling of cells exposed to Nef peptides. The peaks of apoptosis are denoted by Motif 1 and Motif 2. To assess competitive inhibition of Nef secretion by SMR wild-type peptides, culture medium from Jurkat T cells co-transfected with a Nef-GFP clone and various SMR peptides were assayed for exosomal Nef secretion. As shown by GFP fluorescence in the medium, peptides containing one or more wild-type Nef SMR sequence motifs inhibited the secretion of exosomal Nef, while alanine replacement of any amino acid within this region greatly reduced the peptide's effectiveness. This suggests that SMR wild-type peptide inhibits exosomal Nef. An 11 amino acid scrambled version of Nef's apoptotic motif 1, SM1, previously described and obtained from Sigma Genesis, was used as a negative control and had no effect on exosomal Nef secretion. This image of the Kumasi-stained SDS-page gel shows the co-immunoprecipitation of 4 proteins by the SMR wild-type peptide, with 60, 65, 75, and 250 kilodalton sizes. The negative control SMR mutant peptide did not capture these proteins, and these proteins did not non-specifically interact with the affinity resin in the absence of the SMR wild-type peptide. These results suggest that the co-immunoprecipitated proteins were interacting specifically with the SMR motifs of the SMR wild-type peptide. The plate reader assay shows that co-transfection of the anti-mortalin antibody with the Nef expression plasmid completely abolished exosomal Nef secretion. The unrelated antibody had no effect on exosomal Nef secretion. These results suggest that an intact Nef-mortalin interaction is required for exosomal Nef secretion.
In conclusion, after watching this video, you should have developed a good understanding of how to use the described techniques to identify functional motifs in target proteins and to use that data to develop peptide-based inhibitors of these functional motifs. While attempting the co-IP procedure, it will be critical to remember to give careful attention in executing the incubation washes, making sure that the beads are properly agitated during the incubations and that as much liquid as possible is removed from the beads during each wash step. Also, during the cell and lysate resin washes, always allow the resin to settle following the centrifugation, use the narrow end pipette tips, and do 4 washes to adequately reduce the background to acceptable levels.